Alright, so today we're going to talk about DNA structure. But before we know what DNA structure is, we need to understand what DNA itself is. So DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. Alright, so right away that name's going to tell us some stuff. We'll come back to the deoxy part later on. But the first thing that we should notice is that it is a nucleic acid. And we learned earlier this year that a nucleic acid is one of the 4 macromolecules along with lipids, carbs, and proteins. Alright, so what that tells us, since it's a nucleic acid, it's a macromolecule. So in order to figure out where DNA is located, we have to first look at our picture of our cell. So let's pretend like this right here is an animal cell. Right? On the outside of the animal cell, there's a cell membrane. That's how we know it's an animal cell. There's no cell wall. We go into the cell membrane. We travel through. The yellow part would be the cytoplasm. And then in the center of the cell there would be a nucleus. Inside that nucleus is where the DNA is kept. So inside that nucleus, we would see this big giant molecule called DNA. Since it's a molecule, it's not living. Remember, a cell is the smallest living thing. Since DNA is part of a cell, it can't be alive by itself. Now, the reason why DNA is so important is that DNA holds the instructions for how our cells function. So we're going to be able to see that today, how DNA holds those instructions. Since DNA is a macromolecule, it's a polymer. That means it's made up of many individual parts called monomers. The monomers for a nucleic acid is something called a nucleotide. Alright? So DNA is made up of these repeating nucleotides over and over and over again. Just like a train is made up of repeating different types of cars, well, DNA is made up of these repeating different types of nucleotides. All nucleotides have 3 parts. Part number 1 is something called a phosphate group. Phosphate is just an ion. It's an atom of phosphorus attached to some oxygens with a charge to it. Alright? Don't worry too much about that. We're not going to go in super depth about that. But phosphate's just a charge. Charged molecule. So it's just an ion. The second part of a nucleotide is something called a sugar. We're all familiar with it. Well, sugar is just made up of several different atoms. There's a molecule there. And the sugar in DNA is something called deoxyribose. Now, how in the world would you ever remember that name? Deoxyribose? That's hard. Oh, yeah. DNA stands for deoxyribonucleic acid. It's in the name. Deoxyribose is the specific sugar that makes up DNA. You'll notice that it ends in OSE. Well, any word that ends in OSE, when we're talking in biology anyways, is going to be a sugar. So lactose is a sugar in milk. Glucose is a sugar that we've been talking about quite a bit. Fructose, sucrose, those are all sugars that you've heard of before. Later on this year, we'll talk about RNA. RNA, its sugar is going to be ribose instead of deoxyribose. If you think about deoxyribose, we know that if you put D in front of a word, it means without. So like decaffeinated beverage is without caffeine. Deoxy is going to mean without oxygen though. So the difference between deoxyribose and ribose is that deoxyribose is going to be missing an oxygen compared to ribose. So when we learn about RNA later on in the year, it's going to be pretty much the same as DNA, except there's going to be a missing oxygen on the sugar, along with 1 or 2 other little differences. Now, the third part of the nucleotide, so remember nucleotides are always made up of phosphates, sugars. The third part is something called the nitrogen base. Alright, so we'll see that the nitrogen base is the one part of the nucleotide in DNA that can change from nucleotide to nucleotide. So the nitrogen base determines what type of nucleotide we're talking about. Okay, so Like I said, the nitrogen base can change. There's 4 different types of nitrogen bases in DNA. The first of the nitrogen bases is something called guanine. All right, say it after I say it. Guanine. Did you actually say it? You better have. I don't care if you look like a weirdo and your dog's staring at you like you're crazy. These 4 nitrogen bases you're going to be saying a ton. So you don't want to sound goofy when you say them. You want to be saying them right. Guanine. The second nitrogen base is something called adenine. Say it after me. Adenine. The third nitrogen base is something called thymine. Thymine. And the fourth and final nitrogen base is something called cytosine. Cytosine. So together, these 4 nitrogen bases plus phosphate and sugar make up DNA. So remember, each 1 phosphate, 1 sugar, and 1 nitrogen base makes up what we call a nucleotide. Here's a little drawing of a nucleotide for you. So here's 1 nucleotide. You can see here's the sugar, labeled S. That's deoxyribose. There's the phosphate connected to it. And there's the nitrogen base connected to the sugar. That's 1 nucleotide, a sugar, a phosphate, and a nitrogen base. You look, the sugar connects to both the phosphate and the nitrogen base. The phosphate just connects to the sugar, the nitrogen base just connects to the sugar. All right, this specific nitrogen base is adenine because it's A. That's all fine and dandy, but in actuality, DNA is made up of a bunch of nucleotides. So if you look here in this picture, you'll see that DNA is made up of a bunch of repeating nucleotides. There's 1, there's a second, there's a third, there's a 4th. But look, right across from those nucleotides is another nucleotide, a 5th, 6th, 7th, and 8th. That's because DNA is what we call double-stranded, meaning that it lines up in 2 lines with 1 nucleotide across from the other one. What we're going to see is that 2 bases always pair up, adenine and thymine. 2 other bases always pair up, guanine and cytosine. Easy way to remember this, guanine and cytosine are round letters, G and C, so they want to go together. Adenine and thymine are kind of pointy letters, so they like to go together. All right. Down the sides of the molecule, you can see molecule. Did I say mol? Down the sides of the molecule, you can see phosphate, sugar, phosphate, sugar, phosphate, sugar. Other side, phosphate. Sugar, phosphate, sugar, phosphate, sugar, phosphate. So the sides of the molecule are made up of alternating phosphates and sugars. The middle of the molecule is made up of 2 nitrogen bases bonded together. If we there, what we'll see is that DNA kind of looks like a giant ladder with, again, phosphates and sugars on the sides, and then the nitrogen bases in the middle. I forget what I want to do. So a lot of times I'll draw out DNA looking like this, kind of like two combs facing each other, with the sides being assumed that you know that those are phosphate sugar backbones. And then in the middle you have letters. Those letters represent the nitrogen bases. And you'll always notice again that C pairs with G, or cytosine pairs with guanine, and A pairs with T, or adenine pairs with thymine. You may be asking yourself, or maybe not, I don't know, maybe you're asking the dog, that why in the world does C have to pair with G and A have to pair with T? Well, let me tell you. Reason number one why they always have to pair up is based on size. There's two different categories of nitrogen bases based on their size. There's purines and pyrimidines. Adenine and guanine, A and G, are purines. That means simply that they're big nitrogen bases. T and C are pyrimidines. That means that they're small nitrogen bases. Now you should pause and you should be like, what? Why are A and T in separate groups? A is big, T is small? That doesn't make any sense. A and T pair up. Well, take a look at this picture. Maybe that'll help. Let's imagine for a second that two large bases are paired up. That means that the two small bases would pair up. When the two small or short bases tried to pair up, the problem would be they wouldn't touch. The two large bases would make the gap between the two strands too big for the small bases to be touching. So instead, what we have to do is we have to take those two molecules and we have to pair one small with one big. Again, one small, oops, that was really sloppy, with one big. Alright, that keeps the width or the distance between the two strands consistent down the whole entire DNA molecule. So reason number one why they pair is based on size. Reason number two why A and T pair and C and G pair, because if you look at this, we said, I believe that A and G are big or purines, and T and C are small or pyrimidines. So if you look at that, you would think, okay, well then why can't thymine pair with either adenine or guanine? We know thymine always has to pair with adenine. So reason number two why adenine has to pair with thymine, or thymine has to pair with adenine, is based on the number of hydrogen bonds that they form. So these guys form these little attractions between their molecules in the center called hydrogen bonds. What we'll see is that, kind of like pieces to a puzzle, guanine and cytosine both form 3 hydrogen bonds, meaning they're attracted in 3 different places each. When they're attracted in 3 different places each, that makes them go together and fit perfectly. Consequently, adenine and thymine are attracted in only 2 places together. They only form 2 hydrogen bonds. So if you tried to pair together 2 and 3, it wouldn't work. So guanine and cytosine have to pair together because they both form 3 hydrogen bonds. And adenine and thymine form 2 hydrogen bonds, so they have to pair together. So the 2 reasons that the bases pair together, 1 is size, 2 is the number of hydrogen bonds that they form. So here I have drawn one half of the DNA molecule and started drawing the other half. This first half is called the original strand right here, as you can see. The order of those nucleotides that I chose was random. In reality, our DNA could be different from one another, or is different from one another. That's what gives us our differences. All right. Well, this strand over here is going to pair up with the original strand. Oops. This strand over here is called the complement strand. Why is it called the complement strand? Because it complements the first or original strand. So if we're looking at this, we know that adenine pairs up with thymine. Thymine pairs up with adenine. Adenine pairs up with thymine. Cytosine pairs up with guanine. And then adenine pairs up with thymine. So the complement strand for that one would be TATGT. In reality, DNA is super long. One of these guys is called one base pair. Our DNA is about 4 billion base pairs long in humans. That's huge. It's monstrous. If you took it out and unraveled it, it would be almost 4 feet long. That's in one human cell, believe it or not. Right? Well, small strands or small areas of DNA, small pockets or sequences of DNA, nucleotides, determine something we call a gene. Right? And what a gene does is it gives our cells a specific instruction. So you might have a gene that determines your eye color. It gives your cells the specific instructions for how to determine eye color. Your eye color gene might be different than mine. I have brown eyes, so mine might read ATACA. Yours might read something like GGGTCG. All right? That's what gives us our difference. The genes throughout our body. All this is great. All this is awesome. I just want to mention one final thing about it. Kind of seems tacked on here at the end, but there's no better place to put it. We've been talking about DNA as if it was a big long ladder all day. In actuality, DNA is not a ladder. In actuality, it is a double helix. That is a almost spirally type shape where the sides again are the sugar phosphate, and then across the middle would be the nitrogen bases, or the rungs there would be the nitrogen bases. We'll learn more about how Watson and Crick, two scientists, came up with the idea of double helix later on down the road. But for now, what you need to know is that DNA forms this double helix shape. So, quick recap. DNA made up of monomers called nucleotides. Nucleotide has 3 parts: sugar, phosphate, nitrogen base. The sugar is always deoxyribose. The nitrogen base is the one part that can change on the nucleotide. There's 4 of them: adenine, thymine, guanine, and cytosine. A and T pair up. G and C pair up. They pair up because of length and because of number of hydrogen bonds that they can form. DNA forms a double helix. That's about the summary of the lesson. We'll see you next time.